Continuing the chapter on basic genetics, embarrassing moments at gene parties. These are, one assumes, representations of chromosomes. Oh my goodness, Vinny's mutating. This is kind of addresses one of the areas that people get confused on. The difference between genes and chromosomes is that genes are single codes. Chromosomes are very long strings of DNA with lots of codes. Now this is an old overhead, is that any single one of these with labels for various locations for genes already has hundreds, if not thousands, of pictures on them. Is that I like this one because in order to show all the human chromosomes, it just shows a few of the known gene locations at a time probably 30 years ago. But it gives you an idea that there's a lot of stuff here. And you know, you've got a glucose transporter and myosin, one of the main proteins in muscle. Neurofibratomas, a lot of times they name these things after diseases that are connected to the gene not working properly. BRCA1, oh, it's on 17, I thought it was on 14. That's the main breast cancer. Well, the first breast cancer associated gene, collagen and growth hormone. So a bunch of codes for a bunch of different things that do a bunch of different things. And they're linked, but there's no necessity of them to do anything similar to one another. They're just codes. When you need the code to make growth hormone, that's where you go. When you need the code to make collagen, that's where you go. And an individual stretch of DNA can mutate, and if it happens inside a gene, it can change the code, and that's what we're going to talk about next. If I can get any of these things off of here. People working with fruit flies, Drosophila, doing heritability research would find that occasionally a feature would appear in fruit flies that wasn't there before. Suddenly they would, instead of having red eyes, would have white eyes, or all sorts of variations that could be passed on to offspring. Very often recessive, but inheritable. And so in somewhere in the mid 20th century, the idea that heritable features could somehow kind of just spontaneously change, and that the changes themselves could be passed on, was a really big discovery. And so the mutations is the next big thing on the list. Couple of important things associated with that is sometimes the mutation would occur in a single trait. Sometimes the mutation would occur in several traits simultaneously that seem to be unrelated. Now we would look at that and go, okay, is there a single protein serving double, triple, quadruple duty in different systems? So if the protein isn't working, it's messing up a bunch of systems. Or, looking at it with our current knowledge, is the mutation happening to not just a single gene, but a section of a chromosome, and it's affecting several separate genes simultaneously, which is what you get with A lot of times you get damage near the end of a chromosome, and whatever genes are out there are very often completely shut down. What they also discovered, once they knew that this could happen, science people, right? Let's see if we can make this happen. And they discovered that exposing fruit flies to certain types of radiation would increase the number of mutations you would get. That there were certain chemicals that you could expose the fruit flies to, and it would increase the amount of mutations that you get. We now know that ionizing radiation, hefty radiation, X-rays, cosmic rays, gamma rays, can break DNA, and it can break DNA in several places so that it doesn't get repaired properly. And so it can move genes around, it can separate genes out so they don't get passed along when the cells divide. There's a bunch of different ways that radiation can affect mutations. Chemicals the same way. Chemicals that affect DNA can produce mutations. Because we're biologists, I guess. Fruit flies, Drosophila melanogaster is the species name. They're virtually always referred to as Drosophila. It very rarely is italicized. I mean, it's a genus name and it's part of the species. It should be D. melanogaster also as an abbreviation, but it's just one of those things where everybody's kind of used to Drosophila being your basic lab fruit fly. They're little, they reproduce quickly, they're easy to take care of, you don't need a lot of room. Very often, taking genetics classes, you wind up doing fruit fly crosses because it is that easy to deal with them. And something about it along the way, it was like people just forgot to italicize the genus name. So if you see Drosophila and it's not italicized, not necessarily a huge mistake. Neurospora or Neurospora, again, more than one way to pronounce something, is a bread mold. And keep in mind that a lot of people doing research do research on easy to use models in the lab. And Neurospora, through a certain period of the 20th century, was very well studied. And what people were studying were metabolic pathways. What's in play during this process, during respiration in these cells? What's in process during reproduction in these cells? What's in process during protein production in these cells? And trying to pin down, okay, here's a pathway, it's got 5 steps in it. Here's the enzyme for step 1, here's the enzyme for step 2, here's the enzyme for step 3, here's the enzyme for step 4. They picked up some of the information coming from the fruit fly people on mutations. Oh, radiation, chemicals can produce mutations. And they experimented on Neurospora. And what they found was they could produce mutations. They could produce changes in metabolic pathways, where the endpoint didn't happen. But they knew enough about what was going on to be able to go, okay, here's the endpoint. Where along the pathway is the problem happening? And it was different places on different pathways. But what they were finding was that the mutation of the trait was actually a mutation of a single enzyme in the step-by-step process of the pathway. So a mutation was an alteration, and it was changing a single protein. Now, they knew that there were genes here in play. Nobody at that moment had quite pinned down DNA and what DNA did. But now the indication was, from the Neurospora experiments, is that genes were the product of genes were individual proteins because mutations were affecting individual proteins. You change a gene, you don't just change the trait, you change a single protein that causes the trait to happen. Big discovery. And of course, just to bring this back around, most traits are what are called multiple gene traits. If enzyme 1, 2, 3, 4, and 5 are necessary to produce whatever trait you're looking at, there's a gene for enzyme 1, and one for enzyme 2, and one for enzyme 3, one for enzyme 4, enzyme 5. And so the production of the trait requires multiple proteins, each of which has its own genes. So virtually all traits are multiple gene traits. It's just sometimes there's a protein that's so critical to the production of the trait that if you mess it up, you mess the trait up. Now we get to Watson and Crick, sort of. We'll get there in a second. You watch cells divide. You see the chromosomes. Chromosomes do all this fancy dance. There's obviously something going on there. And so for decades, it was like the cells being very careful to move this, to set this stuff up and move it and separate it out into the new cells. We're looking for an information system in a cell that tells it how to do all this stuff. We're looking for the material of genes. Look at these chromosomes. Well, here's the problem with chromosomes. If you take a chromosome and separate it out, and then you analyze it, you've got a handful of different proteins and a nucleic acid that, when you break it down, seems to be a very simple system. Is that you've got a single sugar, you've got a single amino acid, and you've got 4 nucleic acid bases. You got adenine, cytosine, guanine, and thymine. And even then, however much adenine you have the same amount of thymine. However much cytosine you have the same amount of guanine. So people would look at this and go, we're looking for something that could hold all the information to make a cell throughout its entire lifetime, the coding of how to do what it needs to do. This is too simple. Is that there's not enough pieces here to come up with a coding system. Now here's where we get into postmodernism. I got one more thing to do, one more step before we get to the postmodernism part. Back to the list of people were studying phages. Phages is another term for viruses, usually phages that infect prokaryotes. I think it's just a general old term that you could use it for virtually any virus, but if you hear it now, it's pretty much only applied to bacteriophages, phages that infect bacteria. And what people discovered, again, along this continuum of time, a virus would infect the bacterium and turn it into a virus factory. So the virus was getting information into the cell and using the cell's own production machinery to make more viruses. Okay, what's going in? Well, with a lot of bacteriophages, there's a casing, and they interact with the bacterium, and then the casing falls off. And the question is, so, okay, what went in? And the discovery was in a lot of cases with bacteriophages, the only thing that enters the host cell is DNA. Is a little, I don't know if they call it a chromosome, but a little package of viral DNA. And again, that started to underscore maybe it is informational. That's all that's in there, and it's making more viruses. It's making the packaging, it's making the scaffolding, it's making more DNA, and it's releasing viruses by the hundreds or thousands from this host cell. So this was another important discovery, that DNA obviously had to be a coding molecule because phages were using it to make more phages, and it was the only thing they were using. They were using whatever production machinery the host cell had, so maybe there was an interaction that was necessary, or maybe it was converting something, but it was starting to look pretty obvious that the DNA was doing it. Now we get to Watson and Crick. I'll put them here. Watson and Crick get the credit. Wilkins and Franklin probably should get equal credit with them. They helped develop the techniques that allowed them to do what they did. Rosalind Franklin, people say when they give out Nobel Prizes, they only give it out to a maximum of 3 people. And they don't give it to people that are already dead. And Rosalind Franklin was already dead at the time, but she was a woman, and there is some hint that her being Jewish kind of got in the way of her getting credit. Watson, pretty dicey in terms of his attitude toward people of other races and such. But I saw Crick speak once. Really boring guy. But they were the ones that went, okay, if DNA is the coding molecule, how the heck does it work? And now we circle back around to postmodernism. The double helix. They figured out the structure that everybody knows now. It wasn't important what the individual chemicals were. What you were looking at was a sequential molecule. And where's postmodernism come into that? Well, through the course of the 20th century leading up to Watson and Crick was the development of recording technology. Is that originally, grooves in a disc, that one didn't translate quite as well, but when recording technology moved to magnetic medium, first on wires and then on literally rusty tape, is that in order to record stuff, a magnet actually did a very simple change in the sequence of the wire and then the tape going through, and that could be read later in order to hear music or eventually to see images. Complex information could be stored on a simple medium by making it sequential and then reading the sequence. And that's what they figured out was happening with DNA, that the code was in the sequence of one side of this molecule. That helped them figure out how you could make copies, right? We already talked about that. Is it what's on one side, when you're going to make a copy of it, what's on one side determines what used to be on the other side, and now you're going to make a copy of. And so it's called semi-conservative. We talked about that already. And they did eventually figure out the code as a 3-to-1 code, a codon, 3 sequences of nucleic acids, of nucleotides, were what translated into eventually 1 amino acid in the sequence for protein. And they knew proteins were sequences of amino acids. So it's a matter of, once you figure out that you're dealing with a sequential code, you've got a sequential molecule. That's what proteins are. And then it was just a matter of, so is it a 1-to-1 code? Is it a 2-to-1 code? There's some theory that it did start as a 2-to-1 code, but it eventually settled on being a 3-to-1 code. Every 3 nucleotides inside a gene sequence codes for 1 amino acid in the final protein. And we'll find when we get to that chapter, that's a gross oversimplification. They also discovered mutations as changes in the sequence. We'll cover these in details later. This is just kind of an introduction. But making copies, or if you lose something in the sequence, you get what's called a deletion. Okay, so you're reading 3 by 3 by 3 by 3, and then one of your 3 is missing a piece. So now you're starting the next 3 a spot earlier, which changes it. And it changes everything from there on. It is possible to get a nucleotide stuck into the sequence that doesn't belong there. So you're going 3 by 3 by 3 by 3, and now you haven't reached the end of the sequence because you've got an extra one, and that bumps the sequence a different direction. It's called changing the reading frame. If you just swap out the wrong codon, or the wrong nucleotide, then you change that codon. And maybe you change the amino acid, maybe you don't. We'll talk about that later. Often not a huge change in the protein itself, right? Proteins are very long stretches of amino acids. Changing one amino acid to another often doesn't really do anything to the protein. But if you're reading the code and suddenly every amino acid from here on is different because you popped the reading frame, that causes major mutations. Is that the whole sequence of part of the protein is just not what it's supposed to be. So certain, what I call point mutations, mutations of one nucleotide spot, have very different kinds of implications. You can swap things around, you can move them from one place to another. There's a whole bunch of different ways that the DNA can be changed. Oops. Where are we? Okay, I'm going to stick these last 2 things onto this one. It's going to go a little long. In the sequence of history, now we get to genomes, the entire DNA of a particular species. What it really turns out to be is an individual you've chosen from the species. In, I think, the 1980s, the government offered a fairly large prize to the first relatively complete human genome. I think they wanted at least 93%, something like that. But the DNA sequences of human beings, which of course meant a human being that somebody chose to do this on. Determining DNA sequences was very, very time and labor intensive. You needed a bunch of lab techs to analyze even short stretches of DNA. And the government figured, okay, we will give people an incentive to speed this up. And they did it. Is that they automated a bunch of different ways to read genetic sequences, of ways to separate out bits and pieces. A whole bunch of technology was developed very rapidly, much more rapidly than anybody expected. They expected this, I think, to take 50 years. It took about 15. And now the ability to read genomes has gotten pretty good. And so they've done it on multiple humans now. Now they do it on humans in different eras. Is that they do it on old things, which is dicey. The DNA is just not as stable as, when they go, oh, Neanderthal genome. Yeah, some of it. But, they did the Drosophila genome. They did C. elegans genome. They did Arabidopsis genome, is that they've done mice and dogs and bats, well, one species of bat, mosquitoes. Anything of interest, now the tools are there to do entire genomes and then try to parse out, so where's this gene and where's that gene? We've got a sequence from this animal that does this. Tell the computer to look for a homologous sequence in a mosquito. And so having whole genomes has been a humongous advancement. Also, polymerase chain reaction was developed later than this to be able to take a very small piece of DNA and make lots and lots and lots and lots of copies. When we get to the last chapter on DNA technology, we will talk about that. In the current day, epigenetics, to look at regulatory elements, what causes the genes to activate and deactivate, is a big step forward. And also what's called evo-devo, evolutionary and developmental implications of genetics. And that's what we're going to cover in the next part of the chapter. We're going to talk about what's called the modern synthesis, the application of modern genetics understanding to how evolution works.